This module continues our discussion of membrane structure. It looks at the sugar-coated cell surface. We'll look at the glycocalyx, which is really a series of sugars in the form of oligosaccharides bound to proteins, becoming then glycoproteins, that are embedded in the cell membrane. We'll later look at ECM, and we'll see how a common motif of amino acids, RGD, contribute to the formation of the extracellular matrix and its binding to the glycocalyx. We will look a little bit at the roles of some molecules in the glycocalyx and in the ability of cells to recognize and adhere to one another. We'll also take a look at cell junctions, gap junctions, tight junctions, and so on, desmosomes. And we'll look at cancer cells from the point of view of cells being unable to recognize themselves appropriately. So that's the module in a nutshell. Let's define the glycocalyx. It's nothing more in this picture from your textbook than the carbohydrate or glycoprotein layer, most of which is covalently associated with the cell membrane, with the bilipid layer. So you see here integral membrane proteins covalently linked to oligosaccharide sugars. The sugar units are shown in blue. There are also adsorbed glycoproteins that are considered part of the glycocalyx. The adsorbed proteins are not covalently linked and are more easily removed from cell membranes, for example, by heating them a little bit to increase thermal motion. But most of the sugars and proteins are covalently linked to proteins embedded in the membrane, transmembrane proteins. We also see here proteoglycans, which differ from glycoproteins mainly in having longer oligosaccharide chains, but otherwise they're substantively very similar to regular glycoproteins. The glycoproteins and proteoglycans end up on the extracellular surface of cell membranes, accounting for the fact that cells are sugar-coated, and they get these sugars during transit through the endomembrane system, which you may recall includes rough endoplasmic reticulum, the cis and trans Golgi vesicles, and secretion vesicles, as well as lysosomes. But in this case, we'll focus on secretion vesicles at the end of this pathway. Here is basically why cells are sugar-coated. It's a series of processing steps that modify proteins embedded in membranes in the rough endoplasmic reticulum. So here we have an image of endoplasmic reticulum, some ribosomes in the process of producing polypeptides. There's one that's been drawn somewhat more darkly. And here we see glycosylation occurring, the addition of sugar residues added as short oligosaccharides to certain amino acids in the polypeptides, often serines or threonines. These are the amino acids to which one can easily covalently link sugars. Several different kinds of sugars are involved, shown by the different cartoons. They get put on in groups or individually to form what's called a typical core glycoside. When the protein is finished, when the ribosome has finished translating the protein, of course, ends up inside the rough endoplasmic reticulum, and we call that a packaged protein, shown here illustrated in miniature with a core glycoside. On the left now are which sugars are involved: N-acetylglucosamine, sometimes called NAG for short, mannose, and of course glucose. And then other sugars will be added as these proteins progress through the Golgi vesicles and reach eventually their final destination. The additional sugars that are put onto the core glycoside are often referred to as terminal glycosides or terminal sugars. So terminal glycosylation occurs in Golgi vesicles, and here we have an illustration of a Golgi stack within the cytosol of the cell. And in it, in the trans vesicles, it's not labeled here, but the vesicles showing the proteins are trans vesicles, and we see a packaged protein, and we even see a protein that's embedded in the Golgi membrane, having been produced in the RER, but instead of ending up in the RER vesicle space, ends up in the membrane. In the next step that you just saw happening here, let's do that again. A vesicle will bud off of the trans face of the Golgi to form, in this case, a secretion vesicle, which migrates to the cell surface. The membranes fuse. The proteins that are in the lumen come out, and those, of course, are secreted proteins, and the proteins that are embedded in the membrane of this vesicle, with the sugars facing inside the vesicle, become sugars on the cell surface, on the plasma membrane, and this then accounts for why the sugars are only on the outside surface of cells. The so-called packaged proteins, which we say are in the RER, are actually topologically not in the cytosol, so they're outside of the cell. Now let's take a look at the extracellular matrix, ECM, or basal lamina, sometimes called basement membrane or basement lamina. And let's look at some of the molecules that actually attach to the glycocalyx to form the ECM. Some of them you're familiar with. One of them is collagen. It's the main structural protein of cartilage, which is an extracellular material, but it's also present in the ECM. It is so abundant in humans and other animals, it accounts for about 25% of all body protein. We mentioned proteoglycans just a moment ago as glycoproteins in which the oligosaccharide chains are fairly long, and they are part of cartilage as well, and they provide strength, flexibility, and cushioning to cartilage. But again, they are also part of the extracellular matrix. There are a couple of other proteins to mention here, laminin and fibronectin and a few others. I will mention them along the way. And these link components of the extracellular matrix to one another. They may participate in some aspect of cell recognition or cell attachment. Here we have a small electron microscope picture attempting to show you the difference between the glycocalyx, the GC, and the ECM, or basement membrane, or basal lamina. So you see GC is actually pointing to what looks just like the cell membrane around the cell. That's because it is the sugar coating immediately adjacent to the cell. The basement membrane, or ECM, as you can see, can be quite thick. So it's quite a bit of feature superimposed on the glycocalyx. Here's a picture from a different textbook than that you purchased for this course. It shows you a few of those proteins, fibronectin, collagen, laminin, and some proteoglycans are illustrated. Proteoglycans can actually exist free of the membrane, that is, can exist as molecules that are not embedded in the phospholipid bilayer, or they can be part of the phospholipid bilayer. Either way. Collagen is is this yellowish worm-like thing, quite long. It's actually made up of multiple polypeptide strands wound around one another. It has very little three-dimensional structure. It's an elongated protein. The cell membrane, of course, has a lot of transmembrane proteins. Only one kind is shown here, and that's the integrin molecule. Integrin meaning integral membrane protein, but it's actually a receptor that can bind to molecules outside the cell. And in this case, they actually attach to more than one extracellular matrix component. And the question is next, how? How is it possible that the same receptor can bind to not one or two, but in fact to several different kinds of molecules in the environment? The only thing I should add here is what it says on top, and that is the proteins and glycoproteins of the ECM are produced and secreted by the cells on which the ECM will form. By and large, that is the case. Well, the answer to the question, how can integrins bind more than a few ECM proteins and thereby allow the construction of an ECM? The answer is that they have the ability to recognize a tripeptide that is part of larger polypeptides, arginine, glycine, aspartic acid. The one-letter code for those three amino acids is R, G, and D, hence we refer to the RGD motif. And this is a sequence motif that you can find in a lot of proteins in the ECM, and you'll see in a moment a number of blood proteins as well. And we'll see how the RGD works in several ways. Proteins that have this motif include fibronectin, which we just saw in the illustration, fibrinogen, which is a blood protein, laminin, which we saw as part of the extracellular matrix, VWF, or von Willebrand's factor, we didn't see that, that's in the blood, and collagen. Again, RGD then is recognized by integrins, and if you go back to the picture before when you review this, the picture of the ECM, you will see that a fibronectin molecule is bound to integrin, and in another integrin molecule you'll see collagen attached. This is again not from your textbook. It's a larger view of fibronectin. It's a large molecule, but it's a dimer held together by disulfide bridges, which you can see on the right. Each of the cylinders represents a different domain. Each domain has its own function, which means it has motifs for binding one thing or another. So fibronectin can bind heparin, it can bind fibrin, it can bind collagen, and it may have more than one fibrin binding domain, as you can see here. There is a cell binding domain, the blue one, and it can bind to cells because this domain has within it the sequence RGD. And note, by the way, that it has two RGDs. It's the RGD in that domain that attaches to an integrin, which can then help build up the ECM by virtue of its other domains, which bind other components and draw them into constructing an ECM. And here we have an integrin molecule, which you should imagine is attached to the cell membrane, and that's where it's going to bind. It has a region that recognizes RGD. Shown above is a blow-up of the RGD loop embedded in part of the cell binding domain. Fibronectin plays a major role in how cells move on surface. So if we take a bunch of cells and we put them in a petri dish in liquid, and we allow the cells to settle onto the surface of this dish, you can take real-time cinematography, microcinematography, and watch the cells sort of move around in a random fashion. On the other hand, if you take a solution of fibronectin and dip a paintbrush into it and paint a track along the surface in this petri dish, the track can actually direct the cells to migrate in a specific pattern, namely along the track, which is what I've just shown here. This is useful because during development, an extracellular matrix forms along sheets of tissue, and newly differentiated cells can move along this track from wherever they originated to where they have to be before they can differentiate any further. This is very important, obviously, during the formation of an embryo and the development of fetal tissues and so on. Here's another example. Oh, I should have caught that. Another example of extracellular matrix guidance of cell movements. This one occurs in the blood. Neutrophils are white blood cells that migrate between the endothelial cells lining blood vessels to move towards a wound or an infection site. And what's happening in this picture is that these neutrophils, which have specific oligosaccharides in their glycoproteins on their surfaces, recognize receptors called lectins. We saw lectins before. If you remember, lectins are proteins that recognize and bind to specific sugar residues. The lectins on the endothelial cells can bind to the sugars on the neutrophil surface, attaching the neutrophil to the endothelial cell, which then is able to migrate along the endothelial cell and eventually, after several other events, can then insert themselves and squeeze themselves between endothelial cells, actually leaving the blood vessel and going to the site of infection where they can phagocytose or endocytose debris, bacteria, whatever they have an ability to remove from the wound site or site of infection. Again, it's a kind of migration. It involves the neutrophils moving along the basal lamina or ECM of endothelial cells. Let's take another look at a role of RGD motifs in blood coagulation. What you see in the picture are platelets. During blood clotting, they are aggregated by molecules of fibrinogen or fibrin, and they form a meshwork of structure along with the fibrin itself, which forms a meshwork of fibrous molecules to produce a clot, essentially a blockage of blood flow through the vessel. So if you cut yourself, you obviously won't bleed all over the floor for the next 4 hours. Within a few minutes, the blood clots at the wound. Part of that clotting is the aggregation of platelets. It turns out that the fibrin or fibrinogen, either one, has RGD motifs which can bind to integrins on the platelet surface, forming this aggregate that you see in the picture. Now we know that this is what's going on because we can synthesize, make synthetic RGD tripeptides. Now that's just a 3 amino acid polypeptide, a tripeptide, illustrated here as a blue crescent moon. And if you mix those synthetic tripeptides with platelets, what will happen is that the tripeptides will recognize and bind to the integrin. And the result then is that these platelets are blocked from attaching to one another. They can no longer attach to the fibrin molecules or fibrinogen molecules, which you can see floating around in the blue blood here, unattached to the platelet. So these synthetic RGD tripeptides block platelet aggregation by binding to the integrin on the platelets and displacing or preventing fibrin from doing so. So that's an experiment that we did. Not me, but the grand we, did to show that the RGD motif is actually responsible for platelet recognition and aggregation. A last word about integrins. They are transmembrane proteins that can recognize fibronectin or collagen and even several other molecules, but the cytosolic side has other functions. Shown here, and I won't go into great detail, is that the cytosolic end of the integrin is actually articulated with bits of cytoskeleton. We'll talk about the cytoskeleton in detail later, but shown here are some actin molecules and other components of the cytoskeleton attached to the integrin, and some signal molecules that can actually respond when either collagen or fibronectin or other molecules bind to integrin outside the cell. And the response is to transmit information to the nucleus. I won't go into detail here, which then turns genes on or off, getting the cells to behave in certain ways. And this is obviously very important during embryogenesis, when cells have to interact, begin to move, begin to produce new materials. So integrin functions not only to help build the ECM in the first place, but while building the ECM, the integrins allow a cell to respond appropriately during development and in other events. Okay? Now let's look somewhat more deeply at the glycocalyx itself. Cells recognize each other by the structure of their ECM and also by several cell-cell recognition or adhesion molecules that are proteins or glycoproteins embedded in the membrane, that is, proteins that are part of the glycocalyx. But one group of these proteins are called the selectins. They have the word lectin in them, and if you look closely, the selectins, there are a couple of them here, E-selectin, which is found in endothelial cells, P-selectin, which is found in platelets. If you look at the right side of these molecules, you will see that in a moment, in the animation, they are going to bind to glycoproteins in other cells, and here they are. So the selectins use their lectin moiety, their lectin domain, that's the bright green domain at the right end, to recognize oligosaccharide sugars on glycoproteins on other cells. And that then is not only a matter of recognition, but causes the cells to adhere, to stick to one another. This process depends on calcium. Selectins can mediate coagulation or clotting, also inflammation where cells come together during wound healing, of which inflammation is an intimate part. Then we have CAMs, or cell adhesion molecules. The first ones, and perhaps most well known, are neural CAMs, or neural cell adhesion molecules, called NCAMs for short. So we'll take a look at those. And what you saw was the appearance of two of them on two different cells, and they're coming together by virtue of the domain at the end, the amino terminal domains at the end of each. This association doesn't depend on calcium. So neural cell adhesion molecules, as you might guess, will mediate neural connections, that is, the attachment of nerve cells to one another. The domain that actually causes them to attach are found in other kinds of proteins, specifically immune molecules, IgGs, immunoglobulins, which are antibody molecules. It's just interesting, evolutionarily, that immunoglobulins or antibody molecules that have evolved to recognize foreign substances have also been recruited, and perhaps were, even before they were used as immune molecules, were part of cell-cell recognition events, so that IgG domains maybe first were involved in recognizing cells that are like one another, and only later became capable of being modified so that they could recognize foreign substances and deal with them effectively in our immune system. And the last category of molecules I want to talk about briefly are cadherins. They're called cadherins because they involve cell-cell recognition and adhesion. And the C stands for the fact that these are calcium dependent again. And they mediate cell-cell recognition and tissue formation, much like the other ones on this slide. We'll look at the role of cadherins in cell junction. Cell junctions. There are tight junctions, adherence junctions, which we'll talk about by another name in a moment, and desmosomes. Membrane proteins participate in these cell junctions, and you'll see in a moment that cadherins play a major role. Belt desmosomes are the so-called adherence junctions, whereas desmosomes are thought of more like spot rivets. Desmosomes of both sorts are the rivets or glue that hold cells together in a tissue. You can see that desmosomes are created when two cells come close to one another and attach to one another by their cadherins. Tight junctions, if you look at the upper right, are held together by tight junction membrane proteins, or TJMPs, shown here. They are not among the proteins we've talked about so far. And what these will do is create a watertight seal between two cells. And so what you see in the upper right is some dissolved substances above the tight junction membrane protein complexes, but not being able to get through. So you'll see in a moment what good that is. And on the left, I should mention on the left, the other kind of junction is, or the third kind of junction is a gap junction. And these are actually channels which are very small, but will allow, they're hydrophilic, and so they will allow ions to flow between cells. So, adherence junctions, as I said, these are belt desmosomes, and they help to create strong sheets of epithelial cells or endothelial cells that line the blood vessels or your gut, for example. The spot or belt desmosomes, I told you, were rivets or glue that link cells together tightly. The tight junction, made with those TJMPs, form a watertight barrier that can control how substances can move. They can either get into the cell and traverse a sheet of cells from one side to the other by getting into the cell, or they can't get across the sheet. That's what the tight junction is all about. Finally, the gap junction is really less a junction than a communication channel, and it allows ion flow between the cells. These junctions were originally identified by their structures suggested by this electron micrograph, which shows three different kinds. We know that the membrane, all membranes, including the cell membrane cartooned here, are a fluid mosaic which allows lateral movement of components, including proteins, shown here as green and blue things. But cells can, in fact, restrict the mobility of proteins by synthesizing and putting into their membranes barrier proteins, barrier molecules, and collecting certain membrane domains or membrane proteins on one side of the barrier or another. And so in this cartoon, you see that all of the green proteins are above these black barriers and the blue ones below. Some of these black barrier proteins may in fact be cell junction proteins. Mainly, though, they are barriers to the mobility of proteins between domains. They will move within the domain, but not between them. Let's look briefly at the role of cell surfaces in tissue differentiation, which hopefully will tie a few of these pieces together. So here's an undifferentiated cell. Think of it as an early embryonic cell. Illustrated is the glycocalyx as a sort of fuzzy coating of the cell. Remember, these are glycoproteins, also glycolipids, and what makes it a glycocalyx are the carbohydrates that are covalently linked to the proteins embedded in the membrane. During development at some point, integrin will be produced by these cells. The integrin gene will be turned on, the proteins will be synthesized. In fact, they are synthesized in the rough endoplasmic reticulum, and they end up on the surface of the cell. Cells will then migrate and attach at different places in the embryo and begin to differentiate so that you get all the different cell types of an embryo, then a fetus, and eventually a newborn in the case of humans. What is this all about? Well, it's about the cell synthesizing and then secreting basal lamina or ECM proteins, which then bind to integrin and to each other to create the ECM. And it's about the synthesis of cell junction proteins to allow cells, once they recognize one another, to actually form junctions. And so what we see is a couple or three cells here attached to one another and bound to a basement lamina or ECM, shown at the top. Barrier proteins might be synthesized at some point during this process to keep the junction proteins on the sides of the cell, of these epithelial cells, and the glycocalyx proteins that will interact to form an ECM on the upper surface in this case. And here we have the cell junctions, and therefore the cell junction proteins that must have participated in making these junctions, the cadherins and the tight junction membrane proteins, or TJMPs, that we saw named a moment ago. And of course this can produce a very strong sheet of epithelial cells that could line the intestines, line your bronchial passageways, or endothelial cells that line the blood vessels. Finally, normal versus cancer cells. Remember that Normal cells, when placed in culture, will grow to form a confluent single layer of cells, and then they stop growing. They stop growing because when they come in contact, they actually form gap junctions that communicate with one another. And one of the communications is stop dividing, and that's why cells form this sort of flat one-cell layer if they are normal. We know that cancer cells, when they come together and fill the bottom of a plate, continue to grow, eventually filling the entire liquid in the plate. They've lost their capacity for contact inhibition. And it turns out that when you study cancer cells, they don't form gap junctions. They may come in contact, but they don't form the normal communicative junctions that would otherwise tell them to stop growing. When one looks at these cells, it turns out that some cancer cells either have defective integrins, that is, there's a mutation in the integrin gene, and the integrins are no longer capable of binding and forming ECM, or proper cell recognition features, or they actually have defective cadherins, and so they can't form desmosomes or adherence junctions. That brings us to an end of this discussion of this aspect of membrane structure.